Good afternoon. This is now the afternoon session of our CDI Day today, and it's with great pleasure that I introduce our keynote speaker, who's Dr. Bruce Gelb. He is the director and Gogol Family Professor of the Mindich Child Health and Development Institute at the Icahn School of Medicine at Mount Sinai in New York. He's professor of pediatrics, genetics, and genomic sciences. Dr. Gelb obtained his MD from Rochester, completed a pediatric residency at the Babies Hospital of Columbia Presbyterian Medical Center, and then did a pediatric cardiology fellowship at Texas Children's Hospital at the Baylor College of Medicine. He then moved to Mount Sinai and rose through the ranks, being vice chair for research in the Department of Pediatrics before he assumed his present role. He's developed an extensive program in genomics gene discovery for congenital heart disease, and his clinical focus is cardiomyopathy and Noonan syndrome in the past, maybe even now. He directs the cardiovascular genetic program at Mount Sinai. He has multiple models and has used state-of-the-art genomic approaches to study congenital heart disease. He is the principal investigator on 2 R01 grants, 1 R03 grant, a U01, and a T32 grant. He's published over 125 plus peer-reviewed articles and 50 chapters and invited reviews, holds 2 patents, and has mentored many throughout his career. He's also the recipient of multiple awards. Just example is recipient of the E. Mead Johnson Award from the Society for Pediatric Research, the Norman Siegel New Member Outstanding Science Award by the American Pediatric Society. He was elected to the American Society of Clinical Investigation and Institute of Medicine, and he's also a member of the American Board of Pediatrics, Pediatric Cardiology Subboard, Council of American Pediatric Society, and International Pediatric Research Foundation. So the title of his talk today is Genetics of Congenital Heart Disease, Long Day's Journey into Clinic. Dr. Bruce Gelb.
Well, thank you very much, Sharon, for the introduction. Thank you to the organizers for including me in this inaugural and very exciting translational child health research day that you've put together. It's been great so far, and I look forward to the rest as well and hope to do my little part here. So I don't know if this is CME granting, but I don't have any conflicts for this talk. So the issue that I've been struggling with, and others have been, but I have certainly my whole career, is obviously we know the genome's been sequenced, and the question really is, how is that information ultimately going to be relevant for understanding congenital heart disease, and in particular, how is it going to have clinical relevance? So what I hope to do in the time allotted to me here is to tell you a little bit of background about why we think genetics is important for congenital heart disease in the first place, and then move on to do a couple of cases that I think will be obvious to most people, or many people, pretty quickly, but use them as illustrative to how genetics is useful in clinical care, perhaps. And as well, I'll throw in 2 of the studies we've done. One I've done through the consortium, and one I've done in association with the Pediatric Heart Network, and I'll say more about that in a little bit, to show you what we're learning about congenital heart disease genetics at the current time. So by way of background, I think most people will know that congenital heart disease is the commonest birth defect and still accounts for more than its share of mortality. And the number stated here is actually way too low, right? Because if we include bicuspid aortic valve, we get up to more like 2% of the population is born with congenital heart disease. And of course, we know it's a lot higher in babies when they're stillbirth and still higher in spontaneous abortuses, and that's because of, principally because of aneuploidy. And congenital heart disease, in terms of its incidence or prevalence and distribution across lesions, has been studied a lot over time and in many different places. And pretty much Everywhere we've looked, the numbers are quite similar. There are slight differences. There's one little area of Russia where they have a lot of Epstein's anomaly. In the Far East, they have a certain type of VSD. But basically, it's the same everywhere. And we know that ASDs, VSDs, and PDA make up about half the cases. So that's important. The reason I mention that is because that speaks to the fact that there's not likely to be huge environmental factors playing, because you'd see a lot more unevenness across time and place than what we've experienced. I'm going to just very quickly say two things about environment, just so to be complete, because it's not that I don't believe that they have any role. We do know that certain things like maternal infections, maternal diseases as listed here, and certain teratogens are important for congenital heart disease. But at the present time, at least in aggregate, we don't think that's a major impact in causing congenital heart disease. If you think it is important, I might just remind everybody that heart development is really, really early in human gestation and listed here in days. You could add the 2 weeks if you want to do it the way we usually speak of pregnancy. But remember that the heart is largely formed by 7-8 weeks of pregnancy. So we're talking about very early events if we care about exposures. So if you open the textbooks, here's what typically one finds about the impact of genetics of congenital heart disease. We know that somewhere in the order of 5 to 8% of defects are attributable to chromosomal abnormalities. The ones listed here, I'm sure, are familiar to people in the room, and they vary in type and frequency of congenital heart disease, but those are the main players. And then it said that single gene defects account for but 3% of congenital heart disease. So I'm going to try and convince you during the hour that that's probably too low a figure, even though we don't know the right answer at the current time. So when I was a fellow, the model that we lived with was a multifactorial model that included polygenic inheritance, many genes, and some interaction with environmental effects. And that was a model that was put forward in the 1960s by a husband and wife team who happened to be at Baylor long before I was there. But that model started to fade actually as I started fellowship. And the critical finding or study was done by Ruth Whittemore, who was up at Yale, and she was able to do something for the first time that had never been possible before, which is because of the surgery for serious congenital heart disease, we then had women and men, but this was a study of women, who had significant congenital heart disease that were living long enough and were in good enough condition to have children themselves, to ask the question, what was the recurrence risk of congenital heart disease if a mother had congenital heart disease? And they expected the answer to be 1 to 2% or maybe 3%. That's typical for polygenic inheritance. And lo and behold, her study showed that the recurrence risk was 16%. Considerably higher than expected. And for certain heart lesions, such as left ventricular outflow tract obstructive lesions, as an example, you're approaching numbers that are basically Mendelian-like or consistent with a Mendelian inheritance. And very striking is the fact that the lesion concordance, and by that I mean that if the mom, let's say, had tetralogy of Fallot, that her baby had tetralogy of Fallot was about 60%. So everyone saw that this had to be speaking to the genetics. The actual reaction to this piece was disbelief, and so it needed to be replicated, but it has been replicated. And just if some young person wants to pick up a really challenging question, if you do this study over again and ask the question about affected fathers, you get numbers that are a lot lower than this, and that's been replicated too. And why fathers and mothers, why there's sexual dimorphism of that type is not known at the present time. And we don't think it's milieu, but we actually don't know what it is. Okay. One last little bit two last little bits on the epidemiology. So the geneticists in the room will know about something called heritability. It's a complicated concept to explain, but it's a way of asking how genetic is a trait, how much of its variance. It works better for things like height than it does for binary traits. But nonetheless, if you attempt to study heritability, and it's only been done for bicuspid aortic valve and for hypoplastic left heart syndrome, and as you can see here, the heritability is quite high. So again, those studies seem to indicate genetics is key. We have no data, however, about the vast majority of congenital heart disease in terms of heritability. And then the last thing, which is the newest in terms of our understanding of the epidemiology, comes from within the last 5 years, was a study that was done in Denmark. And most people will know that the healthcare system, remarkably in Northern Europe, is actually organized, unlike ours. And as a result, they actually know who has what, who their parents are, who their brothers and sisters are, etc. And as a result, they were able to ask questions like, if somebody has congenital heart disease, what is the risk to their first-degree relative? Something that would be very challenging to do on a population-wide basis in the US. Well, if you look at this column here, you can see that these numbers of relative risk, so for heterotaxy, for instance, is nearly an 80-fold relative risk, to give you an example. And there is no lesion where you're at particularly low risk. It's really quite strikingly high. So I think that was an important finding that they made. So that, broadly speaking, is what we know about congenital heart disease. So when I finished my fellowship, during my fellowship, it was before the human genome had been sequenced, and I began to work on positional cloning, and I got interested in finding genes for congenital heart disease. And so when I got going, first of all, we knew almost no genes for congenital heart disease at that time. And number two, really the only opportunity was to try and study very clearly Mendelian instances of congenital heart disease, which for the most part meant studying syndromic forms of congenital heart disease. So shown here is a list that it probably is not 100% complete. So if your favorite gene that you know about or that your friend cloned is not on there, I apologize. But all the ones that I did are, of course. But so you can see that they have fallen into certain classes, and I want you to remember this. So we hit transcription factors, and of course through the work of the RASopathies, we've hit a lot in signal transduction, but that's important for other things too. And when you add all this up, as much effort, as much fun we've had doing that, it accounts for not so much of congenital heart disease, we need to admit. So the question that I started asking myself along the way was, okay, great, am I like some old English professor, curating this really cool list, but it was never going to have any impact to any person, on the planet. And so how was this all going to make a difference? So that's what I'm going to try and get to. So I'm going to try and make the argument here and now with a couple of cases that the cardiac genetics information is important because in the end of the day, it can improve cardiac care. It improves pediatric care more broadly, and it improves the care that we can provide to families. So I'll give a couple examples of that. So this is a case, a 4-month-old who we saw in the cardiovascular genetics program. She was a female infant who was in heart failure from a non-restrictive VSDN. Even though I'm not trained as a dysmorphologist, I knew that she was dysmorphic when I saw her. And the interesting thing really was her family history. Namely, it turned out that both her sibling and a half-sibling had congenital heart disease. And the other striking thing was that the mom had psychiatric problems. And I didn't get a very good history because they were brought in by a foster mom because their mother had been bouncing out of our hospital with psychiatric admissions. So here is a cardiologist's attempt at a family tree. It's not 3 generations or anything the way geneticists would do it, but it gets the point across. So the child I saw had a VSD. Older sibling, full sibling had tetralogy flow, and then the half sibling had truncus arteriosus. I will say this one had been at my institution, but the other brother hadn't, so we hadn't missed everything. And so, of course, what comes to mind, I imagine right away, is DeGeorge syndrome, but which was described by DeGeorge in Philadelphia in 1965. Of note, he didn't mention the cardiac defects. He was an immunologist and noted the other features listed here. It was pretty clear that the family I was seeing didn't have that. They did not have immunologic problems, and they did not have the calcium problems, and conotruncal defect was not what was seen. Not long after DeGeorge described that, we came to realize that almost all kids with DeGeorge syndrome, of course, have heart defects. It's only a small minority. And this was an interesting list that arrived because some things that are on this, the conotruncal defects, also leave out certain forms of conotruncal defects like double outlet right ventricle, which is illustrative that things are not as we think of them from some of the studies that had been done. Well, so if the family didn't have that, what could they have had? And the other diagnosis that comes to mind is velocardiofacial syndrome. which was described in the Bronx, not far from where I am at Sinai, by Bob Sprinson back in the late 70s. He's an oral pathologist, very much an overlapping, a little bit different heart defects. And then, of course, they have the cleft palate, usually submucosal. They have mild to moderate developmental with speech abnormalities and dysmorphia, as shown here, right? So this is the typical tapered alanase. They tend to have simple overfolded helix, small ear, often attached lobe, although this patient doesn't have that. Well, when we learned about the two syndromes, they overlapped in terms of features, but we came to realize that they overlapped in other ways, that we started to see families where mother, let's say, had velocardiofacial syndrome and the child had DeGeorge syndrome, or there was a lot of heterogeneity in the phenotype. So you guys will probably know that for a long time, we've now switched over, but the test that was used for this was FISH because there was the discovery made of 22q11 deletions, and this is an example of that where this is 22q11, and then it's missing in this particular chromosome. So we now understand that 22q11 deletion syndrome is incredibly important in conotruncal defects. It makes up almost 20% of the cases. This is work done by Betsy Goldmunds at CHOP. And the interesting thing is for some lesions, it makes up a high percentage. And actually, if a child has interrupted arch with abnormal branching of the carotids and the subclavian, then it goes up to 85%, whereas, as I mentioned before, there's very little of some other defects that we used to think of as very much related, like transposition. Okay, so why do we care about this diagnosis? Well, the genetic risks are clear. I think the family I gave gives an example of that. That mom had been missed. She had gone on to have additional children who were at risk, and she was certainly unaware of that. And that's an opportunity that we have to take care of. There are enormous medical risks for these kids that we have to take care of, and that's why it's important to know about these. The speech and language delay, almost all of them will have, and we heard a talk earlier about the importance of early intervention and surveillance of developmental. This is a group you can be on the lookout for. These kids have a particular finding in their carotids. They're medially displaced. So when and if they need palatal surgery because they have nasolalia, the ENT, unaware of the 22q11 deletion, can easily wind up in the carotid while operating. So that's important to know about. And hypocalcemia, while we think of it as a newborn problem, actually can pop up later. And then these kids have late onset psychiatric disorders that the pediatricians have to look out for. And then from the cardiac point of view, first of all, there's risks such as a blood transfusion and graft versus host disease, or immunologic deficiency with an open chest. And we lost a patient to this with truncus at my institution a bunch of years ago. But we also now know that the outcomes depend on this. So the kids with tetralogy of Fallot with 22q11 have worse outcomes, even with the same anatomy, than kids without it. Well, it turns out this is a pretty important problem. Its frequency is 1 in 4,000, and about 10% of the cases are inherited. And we now know that the difference between patients is not because the deletion, because it's stable from parent to child. And so we actually don't yet know. And an ongoing important area of research is to try and figure out whether it's alleles on the existing copy of chromosome 22 or whether it's alleles elsewhere in the genome. And there's a lot being done on that. So why does this keep happening? Because most of it is de novo. And the answer turns out to be one of the lessons we've learned from the Human Genome Project, and that is the importance of flanking DNA sequences. So shown here is the 22q11 region, and this was work that was done in Bernice Morrow's lab up at Einstein. It was done by Lisa Edelman, who's now at my institution. And basically, this is one of several places in our genomes, there are many of them actually, where there are identical sequences flanking, this is the central area, and flanking it, this region and this region are very, very similar, nearly identical in sequence. And these are referred to as low copy number repeats, or LCRs. And this is the only place in the genome where these sequences exist. They're not spread out. Other regions have their own unique sequences. And the problem with this is that Mother Nature has, in essence, set a trap for us. And here's the problem. So during meiosis, as you remember, there are crossing over events between sister chromatids. And ideally, this is supposed to be the LCRs lining up correctly. They would line up correctly. And so no matter how the crossover would occur, you wind up with the normal situation. However, sometimes those sister chromatids do not line up correctly because the identical or nearly identical sequences find each other even though it's improper, and that's why you get non-allelic homologous recombination. If the crossover event goes in this direction, you get what we have recognized as 22q11 deletion syndrome. And you might be thinking immediately, okay, well, what happens if it goes the other way? You should wind up with an extra copy, 22q11 gain. And it turns out, now that we're scanning, we know that there are patients that have those as well. They tend to be subtler than the deletions, which is why we didn't describe them as a syndrome. Well, 22q11 deletion syndrome was described as I was a fellow, so in the late 80s, early 90s. And we would nowadays refer that as a copy number variation. And this was one of the lessons that emerged from the Human Genome, another lesson that emerged from the Human Genome Project. And that is that there are these structural variations in our genomes, and everybody in this room has these abnormalities in copy number. They can be gains or they can be losses. They can vary in size from quite small up to several million base pairs of DNA. About 12% of the human genome is in play, and again, all of us have them. And most of them, as far as we know, are benign polymorphisms that are not responsible for disease. But then there are many other CNVs that are important that certainly cause disease, and they've been associated with neurologic disorders. Well, we now have technology to readily detect them, and just in case people haven't seen it, the one that is used, at least in our shop, there are other technologies to use, but just as an example, is comparative genomic hybridization, where you do fluorescent labeling of patient and a pool of controls, and then you competitively hybridize them. And for the example here, I've just made 3 spots from chromosome 1, 21, and 22q11. And when you do the competitive hybridization, they should equally hybridize, resulting in the color that's intermediate. If, however, you have an individual who's got an extra copy, as in trisomy 21, you can readily see that because the color shifts from the yellow towards the red because it's a 3-to-2 ratio rather than a 2-to-2 ratio. And conversely, if your patient had 22q11 deletion, you can see it shift the other way because now you have a 1-to-2 ratio. So there you go, it shifts towards the control. Well, the beauty of this test is you can test anywhere in the genome. So whereas before we had to ask a specific question, we have to say, oh, I think this patient might have 22q11 deletion syndrome and go test for that with a specific test. Now we can do the agnostic test and detect these anywhere. So how are we using these in cardiovascular genetics? So I'll give you an example of a patient that we saw in our program. It was a 16-year-old girl who came in. She had a secundum ASD, and she had learning issues. I would say they were on the mild side. The truth is we kind of missed it at first in the program because she was bilingual in Chinese and in English. So, it seemed like how bad could that be? I don't speak Chinese, for instance. But it turned out she was in fact struggling. She had struggled before she had immigrated, and she was struggling in New York. She had no discernible dysmorphia. And she had no other major malformations. We went on and did the tests I just described, and it turned out she had a de novo. Once we tested her parents, we discovered it was de novo deletion of 1.5 million base pairs at the transcription factor GATA4. Now, we know the point mutations in GATA4 cause ASDs, and their loss of function. So there's no doubt that that causes secundum ASDs. And we now know well that these larger deletions are responsible for learning problems too. And I could give other examples like that as well. So that led to the research study I wanted to present. So one of the things I've been wondering about is how important are the genetic findings for outcomes in a broader sense. So I had an opportunity to do that by studying 2 groups of patients who had been in clinical trials. So the Pediatric Heart Network, for those who don't know, is an NHLBI-funded consortium that has been doing clinical trials. So 2 of the trials that they did, one is called the Infants with Single Ventricle Study. This was a study that was using enalapril versus placebo, trying to improve outcome from heart failure, for which they used weight for age Z-score as their marker at 14 months, and they had a number of secondary endpoints as well. And their bottom line conclusion was that it was not efficacious. And a related trial was the single ventricle reconstruction study. This was a randomized clinical trial, which is an amazing thing, by the way, that you could get a group of surgeons to agree randomly to do one operation or another for hypoplastic left heart, but that's what they did. And they either had the kids undergo Norwood with a Blalock-Taussig shunt, which was the traditional way of doing it, or the newer way, which is a right ventricle to pulmonary artery conduit, often referred to as the Sano modification. And the primary outcome there was transplant-free survival at 12 months. The primary endpoint was met, that the Sano was better than the traditional, the Blalock-Taussig shunt. We've been seeing ongoing data from this group, and now further out, those differences have disappeared. So actually, the two surgeries are approximately equal. So I was able to get an ancillary study of the patients in this, and the rationale was to see how prevalent were CNVs amongst individuals who had these type of heart defects. But more importantly, because a lot of people were starting to look at this question, was to ask the question, how much do CNVs affect the outcomes? And the reason I wanted to do that was it was my belief that this might become important for design of clinical trials, but also for clinical care. So our hypothesis, of course, was that pathological CNVs would affect outcomes for kids with congenital heart disease. So I feel like I went through. So we studied about, I think, 225 or so children who had been in these studies. Unfortunately for us, they had not enrolled when they had enrolled every child, they didn't get DNA because genetics wasn't on their radar screen. So they wound up getting the DNA at 15 months of age. Their 1-year mortality in the surgical trial, I think, was 30%. And so understand that if CNVs, for instance, are important in driving mortality early, we wouldn't have detected that in this particular study. So that's a limitation of what we did. And I won't, for the interest of time, but if anybody's interested, they ask me later, how we define CNVs as pathologic. It's a not trivial issue how you know if they're pathologic or not, but we did our best with that. I'll suffice it here to say that we tried to look at bigger ones. So these were CNVs that were rare and 300 kb, 300,000 base pairs or larger. So what did we find? We found in our cases that 14% had those sorts of CNVs that we deemed likely pathologic. In contrast, in the control group, we found only 4%. So obviously there's 10% excess, which was a significant amount. The ratio of gains to losses was quite similar, 4-to-1, 3-to-1. And the sizes of the gains was very much the same, but of the losses, you can see very much larger in the cases than in controls. Well, then we noticed that amongst the numbers that we had 13 CNVs that had been seen before and were known to cause genomic disorders. So those are listed here, and for people who are in genetics, you will recognize, I already mentioned GATA4, but some of them, such as the 1q21 and some of the ones on chromosome 16 are well known, and they've been associated not just with congenital heart disease, but with intellectual and developmental delays, and they've been associated with psychiatric problems as well. So they don't, for the most part, cause traditional syndromes. There's no obvious syndrome associated with them, but they've been associated with, for lack of a better term, badness. They've been associated with autism, with schizophrenia, with cleft palate, and with congenital heart disease. So these were clearly and definitely, and made up about 5% of cases. These were clear. So then we went on to look at the outcomes, and this is the reason we had done the study. The two studies had very carefully defined the outcomes for this patient group, and in particular, they had administered the Bayley scales of infant development at age 14 months. So we had their mental developmental and psychomotor developmental indices scores available. We also had careful measurement of height, weight, and head circumference for this population. So if we just start with the question, those that did not have a CNV than those that did, the only difference we found were that there was poorer growth in those that had CNVs. When we broke it down, and admittedly the numbers are getting small, particularly for the lost CNVs, we saw in the lost CNVs that the neurocognitive development was clearly appearing to be worse. Again, this is not surprising. In general, in genetics, lost CNVs are more deleterious than gained CNVs. And then if we looked at that group I just told you, where we knew that these were causing disease and associated with genomic disorders, you can see all the indices and growth are really very far off. So this was clearly a driver of bad outcomes in this population. So the next thing we did was not my idea, it was Jane Newberger's idea. She's a researcher, a very good researcher up at Boston Children's. And she is the PI for the extension phase of the surgical trial. And she said, Bruce, we are now having all the kids who are surviving be examined by a clinical geneticist. And that person at whatever center is deciding whether or not the person has a clear genetic disorder. It's a lot easier to tell now that they're older, and or whether they have some form of mild dysmorphia. So of the kids that we had examined, about half of them had survived and had been examined by the geneticist. And of those, roughly 30%, 33 of them, were deemed abnormal by the geneticist. 4 had known disorders that they were diagnosed with, and 29 of them were discovered to have dysmorphic features. Well, how about those in this cohort who had CNVs? So it turned out that of these, 14 had CNVs. And if I had one of those clickers that you guys could vote, I think you would vote that we're going to see that there should be an enrichment for these things. That is not what we found. None had been diagnosed with a genetic syndrome. Only 2, which is certainly not 2 out of 14 is not more than 29 out of 116. So only 2 had dysmorphic features. And so net, no insult to my genetics colleagues, but the sensitivity of a good clinical genetics exam in discovering these is 14%. So not very sensitive at all. And then we went on, now that this is the best we could, to try and break the patients down in terms of outcomes. So if we took the kids who did not have a CNV, did not have a syndrome, and didn't have any dysmorphic features, these are their outcomes shown here. It's below population norms. The 100 is normative for both of these indices, and obviously a z-score of 0 should have been the norm. If they just had mild dysmorphic features, interestingly enough, their outcomes are no different than those that had truly nothing. However, those that had the pathologic CNV and or had a syndrome clearly had much worse outcomes. So I think this was pretty strong in saying that CNVs are a driver of outcomes in this population. I would argue that on this basis, this just came up for us actually. I mean, all these kids with hypoplastic left heart and related, I think it's reasonable to go looking for these defects. Whether, for instance, it's as true amongst kids with conotruncal or not, I don't know. Conotruncal, you're going to probably do it anyway to look for 22q11. How about if you have a kid with still some other lesion? I don't know what. You name whichever one you want. I don't know the answer. We'll find out in the fullness of time. But I'll be surprised if we don't find that most all of them, the CNVs, are really a driver of outcomes. And I would hope in the future that will also be taken into study design for clinical trials. I think it will. Okay. A second family, I promised 2 cases. So this was actually the first family we ever saw in the program back when we got it going in the early 90s. Truth be told, these guys weren't even alive, I think, even though I've indicated he's the propositor. So this was a pretty striking family. This boy has tetralogy of Fallot. His older brother has tetralogy of Fallot. He has one sister with no cardiovascular abnormalities. He has 2 siblings with peripheral pulmonic stenosis. Obviously, that's a normal finding as a newborn often, but is not normal after a few months of age, and these were much older than that. In one case, it was mild. In one case, it was severe. And interestingly enough, the mom had mild peripheral pulmonic stenosis as well. So the one disorder that comes to mind with that combination that I would have thought of was ALAGILLE syndrome, also known, of course, as arteriohepatic dysplasia. And ALAGILLE, when he described the syndrome, described the complete syndrome, which was based on neonatal jaundice due to paucity of intrahepatic bile ducts, and then recognized involvement of 3 other systems, that there was the congenital heart defects, including peripheral pulmonic stenosis. There are the eye defects, mostly of the anterior chamber of the eye, vertebral abnormalities, particularly butterfly vertebrae, and then there are typical facial features that I'm going to show you in a moment. And ALAGILLE recognized that there were some individuals who had the neonatal jaundice and so on, and only had involvement of some of the other systems. So that was, when I was in training, that was referred to as partial ALAGILLE syndrome. Well, none of this family had had jaundice as a newborn, so clearly they did not meet these criteria. So shown here are facial features. Typical thing are sort of triangular faces, deep set eyes, some hypoplasia as shown here. Some individuals, such as this one on the left, I think are more striking than others. Here's another little one with it. I mean, jaundice is obvious, but I wouldn't say that the dysmorphia is like so very striking, at least for me as a cardiologist. This is a dominant disorder. It's highly penetrant, and however, it's quite variable, so it's not that unusual to make the diagnosis in a child and then realize that a parent has it. However, like most autosomal dominant disorders, 30 to 50% are sporadic cases due to de novo mutations. And a locus for this was first identified through a chromosomal abnormality on chromosome 20. And then Nancy Spinner's group at CHOP went on to identify first jagged 1, which accounts for most cases, and then notch 2. And these are point mutations for the most part that cause this. Well, indeed, when we worked up our family further, we noted that the boy I showed you had normal LFTs. He had a normal spine. To our embarrassment, when we went back and looked at the x-rays from his brother who had had tetralogy of Fallot repair at our institution, he had normal LFTs, but on the x-ray, he had butterfly vertebra that we really hadn't noticed in real time. A little bit embarrassing. And then when I drew blood on mom, she had elevated LFTs on one visit and normal LFTs on a subsequent one, so very mild liver involvement. And one of the children came back for repeat heart surgery. This is now probably about 3 or 4 years ago. And so I inquired how the others in the family were doing, and I learned that the mom had died. And when I asked what happened, it turned out she had died from a ruptured cerebral aneurysm. And that was just about as we were learning that cerebral aneurysms are part of the phenotype of this particular disorder. And in fact, when we went ahead to study them with Nancy Spinner, it turned out that this family has a classic ALAGILLE causing a mutation that causes a frame shift because it's a 2 base pair deletion. So why do we care about this one? Again, there's the genetic risk. There are parents with partial phenotypes, as in our family. There's a lot of different things that we have to surveil for, including developmental delay, which was mild, which I think some people in this family turned out to have. And from a cardiovascular point of view, the multi-system involvement is certainly important when undertaking surgery, particularly the liver disease and the kidney disease. And then we have to surveil for the aneurysms. And truth is, we don't know exactly how to do this the right way. We know how to image, we just don't know the frequency and what to do when they're negative. Which leads me to the next study that we've been involved with. So I told you that we've been studying families where we had clear Mendelian disease. And about 6 years ago, through the leadership of Gail Pearson, who's sort of our white knight at NHLBI, she's a pediatric cardiologist, she funded, she had already put together the Pediatric Heart Network, which I mentioned, and she got NHLBI to invest in 2 other major research consortia. One of which I'm not a participant in, I'm not going to talk about, is called the Cardiovascular Development Consortium, and it's 5 centers, and they're doing really quite remarkable work around cardiac development. But the other major consortium, which I am a member of, or my institution is a member of, is the Pediatric Cardiac Genomics Consortium, or PCGC. So it is 5 sites, and that includes the Harvard, two institutions at Harvard, CHOP, Columbia, my institution, and Yale. The clustering geographically was accidental. And the purpose of this was to try and figure out the genetics of congenital heart disease. So I'm relatively pleased that we didn't film our most initial meetings when we had arguments about what we were going to do and how we were going to do it, because the truth is we felt very much, and even to this point, felt like we're stumbling around in the dark. The fundamental problem is, although we know that congenital heart disease is genetic, and I laid out that evidence for you at the beginning, we don't know the architecture of congenital heart disease. That is to say, we don't know whether it's polygenic, oligogenetic, whether there's a lot of autosomal recessive disease. These are things that are largely unknown. And of course, you would want to know that to figure out how to do your study design. So understand that we've done this without knowing. So here's how we've organized ourselves. We have the sites where we're all recruiting, and we've also, many of us, have brought in ancillary sites that participate as well. So for instance, Cohen's Children's Hospital at LIJ North Shore is an ancillary site for our site. And then we've been recruiting as best we can individuals, adults or children who have congenital heart disease in order to get genomic DNA. For some, we've been getting tissue. And then we've been banking that at Coriell. We've been getting the nucleic acids. And then we set up a series of cores to do various genomic things. And most recently, we set up an IPS core as well, which is at my institution. And then downstream of the data generation, we have a data hub that's been put together. It's fairly complicated that Pete White headed up at CHOP. And then the data goes back out for analysis. So it's a somewhat complicated and bulky design. The files for exome sequencing, we heard talk about that this morning, are quite large. So moving these things around is not a trivial operation. So this is one sign of success. So you probably know that if you look at most NIH clinical endeavors and ask how are they doing with recruitment, they're usually falling behind and explaining why they've fallen behind. These were the numbers that were projected for us from 25 to 100%, and we've actually been recruiting above the 100% target all the way through. So these data are not quite up to, we're almost up to June. We will be, I think, just about at 8,000 individuals with congenital heart disease recruited by this June. That's quite a large collection. These are the characteristics of the individuals. I don't think, they are not the complete spectrum of congenital heart disease. We have focused more on more severe things, so definitely an overrepresentation of conotruncal defects and LVOTOs and heterotaxy. So for instance, ASD and VSD, which you know make up a lot of congenital heart disease, make up only about 10% of our collection. We also seem to have an enrichment of kids that have other problems, 40%, and a very small percentage are already carrying genetic diagnoses. For 40% of the individuals, we have both the mom and the dad DNA for those individuals as well. And then there's about another 20-30% where we have one parent, usually the mother. So the first major study that we undertook was one using whole exome sequencing. We heard about what the exome is this morning, so I won't reiterate that. And our thinking in doing this was that we knew that so much of congenital heart disease appeared to be sporadic. And we knew that there was poor reproductive fitness evolutionarily, so we wouldn't expect common variants to play a huge role in congenital heart disease. And therefore, the hypothesis that we had was that de novo point mutation and insertion deletion, so-called indels, would underlie some significant proportion of congenital heart disease. So we went ahead and we did exome sequencing on 362 trios. And it was a mix of patients with mostly hypoplastic left heart syndrome, tetralogy of Fallot, and heterotaxy. And what we expected was not what we found, of course. So we expected that when I put up this slide, I would show a number here that was a lot larger than that number there. So, but that is not the case. So it turns out that the number of de novos in the cases and the number of de novos per trio in the controls was identical. And just a quick word, if you're wondering what we did for controls, we were actually we didn't think out our study very well, is the truth. We went ahead and did the cases without thinking about the controls, and then we got lucky. It turned out that Yale, where we did our exome sequencing, was simultaneously doing a study of autism. And the autism group was a lot more savvy than us. So rather than do trios, they did quads. So they did two parents, a child with autism and a sibling without autism. So we grabbed the data from the parents and the sibling who did not have autism and called that population normal, which I think is a reasonable thing to do, just so you know. So this is obviously not significant. And I should also say, if anybody cares about details, these numbers are a little low. Many people would say you should file 1.2, and there are technical reasons for that we know. So we're probably leaving some de novos on the table, so to speak. So I could explain that later if anybody cares. So that was very frustrating. We went on then to say, well, maybe the shape, the distribution is different. So for instance, we looked at age effect in parents. We didn't see any difference between cases and controls. And then we said, well, maybe in congenital heart disease, it's lumpier. And there are some kids that have 3 or 4 de novos and some kids have 0, whereas it's more of a Gaussian curve in the controls. Well, that turns out not to be true either. So finally, we had what turned out to be the answer that helped solve the problem, we went ahead and dichotomized genes according to how they were expressed during cardiac development. Obviously, we didn't have data for human cardiac development. We had mouse cardiac development, and these were RNA-seq data that had been generated from embryonic day 14 and a half. And I will say that we also looked at similar data from embryonic day 9.5 and get basically the same results. I'm not going to present those. And then what we did is we took the genes from the RNA-seq set that had orthologs in human exome, because those are obviously the only ones we could deal with. So of the 20,000 or so, you're left with nearly 17,000 genes. And then we divided them into those that were highly expressed during cardiac development, which we refer to as high heart expression, or HHE, and the lower 3/4 were the low heart expression, or LHE. I should also say that we reanalyzed our data a few different times using 20%, 80%, 30%, 70%, and pretty much no matter what you do, you get results that are pretty similar to what I'm going to show you. So we're not cherry picking. You'll have to take my word for it. And then anyway, having done that, we then reanalyzed to say, is there a burden of mutations amongst the genes that are more highly expressed during cardiac development, obviously as a surrogate for saying they're important for cardiac development. So here's what we found. It's a little bit complicated, so I'll take a moment to walk through it. So shown in red are the congenital heart disease versus control for HHE, and in blue is for LHE, same comparison. So these are all de novos. So this is HHE, and you can see there's you're significantly above 1 in terms of ratio of cases to controls, whereas amongst the LHE genes, you don't see that at all. It's a mild effect, admittedly, borderline. And part of the reason it's likely to be borderline is we included all de novo changes. So that would include, for instance, silent or synonymous changes where we know the protein isn't changing. And while there are mechanisms whereby synonymous changes can cause disease, for the most part they don't. And so if you filter those away, you can see that you get a more significant result and your odds ratio goes up. And then if you filter away alterations of amino acids at positions that are not evolutionarily conserved, which tends to be a marker for things that would be tolerated if they change, you can see that it goes up still further. And finally, if you're left with just the damaging ones, so those would be nonsense mutations and frameshift mutations, you can see that we have an odds ratio in excess of 6 and a very significant p-value as well. So that was very gratifying. And then the next question we looked at was, is this true for all of congenital heart disease, but is it true as you look at the various lesions? And the answer turns out to be, while the numbers are different, this is a small group, so this may be biased, but basically we were seeing these changes across all forms of congenital heart disease that we looked at. So then the next question we wanted to ask is, well, are these de novo changes altering a particular or more than one biological process? And the answer turned out to be there was only one when we did that analysis that met significance after accounting for multiple hypothesis testing, and that was things that altered methylation of histone 3 at lysine 4, H3K4 methylation. We observed 8 changes out of the 249 protein altering changes, de novo changes, whereas you would have expected 1 based on the number of genes and so on in that group. Amongst the controls, which were a roughly similar sized cohort, for example, we saw none. Second reason that we thought this was likely to be real is that 3 of the genes that were altered were already known to be associated with congenital heart disease. So MLO2 mutations cause Kabuki syndrome, CHD7 mutations cause CHARGE syndrome, and KDM6A are a less frequent cause of Kabuki syndrome as well. So that was also proof of principle. You might guess, since histone methylation is such a fundamental process, you might expect that these individuals would tend to have extracardiac manifestations, and almost all of them did. So that was also sensible. We only saw 1 gene in our set of this experiment that we hit more than once. That was SMAD2. The likelihood, given the number of de novos we had, of hitting any gene twice was quite low. So this actually is significant that you hit it twice. And it's interesting in that SMAD2, its protein is downstream of something called nodal, which is important for organization of the body's left-right axis. And you hopefully know that in heterotaxy, it's that left-right axis that is disrupted. And it turns out that SMAD2 is also important for histone methylation. It's relevant for H3 K27 monomethylation demethylation. And interestingly enough, both of the children who had these mutations had congenital heart disease that is the type you would tend to see in heterotaxy, even though they didn't have heterotaxy. So their abdominal situs was normal, but they certainly had congenital heart disease consistent with heterotaxy type disease. So the conclusion of this study, and this is going to be where I'm going to finish up, is that de novo mutations appear to be non-randomly distributed amongst genes that are more highly expressed in the heart during cardiac development, again, as a marker of those that are important for cardiac development. And we've clearly implicated chromatin remodeling in that process. It looks like these de novo mutations account for something like 10% of congenital heart disease. And we would consider that a minimal estimate. Remember, I told you that because of the study design, we had to be relatively strict about our calling, and we know that there's some other de novos that were left out of our calling parameter, maybe up to a third. So it could be still higher. And the interesting thing is there are methods for taking our results and extrapolating to how many genes there are likely to be based on what we found. And although the range on this is, the confidence limit is very broad, the fact is it appears, as one might have guessed, and as we heard about for autism earlier, that congenital heart disease is going to be highly, highly heterogeneous genetically. So if there's anybody out there is thinking of getting involved and wondering whether there'll be anything for them to do, I would just encourage you by saying we are going to be busy for endless decades trying to work through what these genes are and what they do, and then think of ways to deal with them and make that information useful. And I'll just tell you that we've now gone on, we don't have data yet, but the good thing about hitting H3K4 methylation is that you can actually study that in vitro. And so we've now gone on to make iPS cells from patients with these defects and are beginning to study how cardiomyogenesis and myocytogenesis is altered when you have abnormalities in H3K4 methylation. So this, for those that don't recognize it, is Heart Mountain. It's called Heart Mountain because it's got what appears to be a heart sort of naturally carved into its side. And I wouldn't say that those of us in the field that we've quite summited yet, but I think, we were down here at base camp when I finished fellowship, and I'd like to believe we've made a little progress. And I want to thank you for your attention. Thanks.